Hello everyone and welcome to my YouTube channel Perfect Lab. So before bacteria can be tested against antibiotics in the laboratory, the bacterial suspension must contain the correct number of cells. And if there are too many or too few bacterial cells, the test result may be inaccurate. So this is where the McFarland standard comes in. And the McFarland standard is a simple but essential tool used to standardize bacterial density before antimicrobial susceptibility testing. So in this video, we will explore what it is, why it's important, and how it's used in the laboratory. So let's get started. So we define the McFarland standard as a turbidity standard which is used in microbiology to estimate the concentration of bacterial cells in a suspension, and this is done to ensure that the bacterial inoculum, which is used in tests such as antimicrobial susceptibility testing, is standardized in order to produce accurate and reproducible results. Or we can also define the McFarland standard as a reference suspension with a known turbidity that is compared with a bacterial suspension. And what we have to notice is that the bacterial suspension is always adjusted until its cloudiness matches the McFarland standard to indicate a specific bacterial concentration. So for this picture over here, and basically in antimicrobial susceptibility testing, we don't just use any McFarland standard but 0.5 McFarland standard. And if you want to perform AST on our isolated organisms, we will have to adjust the bacterial suspension until its cloudiness matches with the 0.5 McFarland standard over here before running our test. Why standardizing bacterial suspension with McFarland standard is important in antibiotic susceptibility testing. So standardizing the inoculum is essential because the number of bacterial cells in suspension can significantly affect laboratory results. So example, so if too many bacterial cells in suspension is used in the antibiotic susceptibility testing, the antibiotics may appear less effective to result in smaller zones of inhibition, which may be a false result. And also, if too few bacterial cells in suspension is being used, the antibiotics may appear more effective to result in larger zones of inhibition, which is also a false result. So it is always important to standardize our bacterial suspension with the correct McFarland standard, and in this case 0.5 McFarland standard to ensure reliable and accurate results in antimicrobial susceptibility testing. So now, based on this information or knowledge, let us now see the importance of using the correct McFarland standard, and in this case 0.5 McFarland standard in antibiotic susceptibility testing in the Kirby Bauer disc diffusion test by 1. Using a bacterial suspension that is more turbid than the 0.5 McFarland standard, and 2. Using the bacterial suspension that has the same turbidity as the 0.5 McFarland standard, and finally using a bacterial suspension whose turbidity is less than that of the 0.5 McFarland standard. So with the first scenario, where a bacterial suspension with higher turbidity than the 0.5 McFarland standard is used, what it means is that there are too many bacterial cells in this suspension, and as a result, the bacterial concentration is higher than the 0.5 McFarland standard, which has a bacterial approximatory concentration of 1.5 times 10 exponent 8 CFU per ml. So when this bacterial suspension is used in performing AST in the Kirby-Bauer disc diffusion test. This heavy inoculum will make the bacteria appear less susceptible to antibiotics. This is because there are more bacterial cells for the antibiotics to inhibit, and as a result, the zone of inhibition becomes smaller, which may falsely suggest that the bacteria are resistant, whilst in actual fact, they may not be resistant to these antibiotics. So let's look at the third scenario before coming back to the second scenario. So in the third scenario, it involves using a bacterial suspension that has a less turbidity than that of the 0.5 McFarland standard. And what it means is that this suspension has too few bacterial cells in suspension, and the bacterial density or concentration is lower than that of the 0.5 McFarland standard, which has a bacterial concentration approximately 1.5 times 10 exponent 8 CFU per mil. So whenever this bacterial suspension with less bacterial cells or inoculum is used in antibiotic susceptibility testing, because there are fewer bacterial cells, these antibiotics can stop their growth easily, and as a result, make the bacteria appear more susceptible to the antibiotics than they actually are. And these may falsely suggest that the bacteria are highly susceptible, whilst in actual fact, they are not. So the second scenario, which involves adjusting the turbidity of the bacterial suspension to that of the 0.5 McFarland standard, is the way of standardizing our bacterial suspension before performing our antibiotic susceptibility testing in the Kirby-Bauer disc diffusion test. And what it means is that the bacterial density or concentration in this suspension is the same as that of the 0.5 McFarland standard. So standardizing the bacterial suspension to the recommended turbidity will always ensure that the test starts with approximately the same number of bacterial cells as that of the 0.5 McFarland standard. And as a result, this produces a more accurate and reliable antibiotic susceptibility result, particularly in the Kirby-Bauer disc diffusion test, to allow results to be compared confidently between laboratories. So, in summary, using the correct 0.5 McFarland standard in antibiotic susceptibility testing ensures reliable and reproducible results, especially in the Kirby-Bauer disc diffusion test. And the McFarland or the 0.5 McFarland standard in AST helps to standardize the number of bacteria in each test, and it also allows accurate comparison of results to support proper interpretation and clinical decision making. Let's look at the composition of the McFarland standard. So McFarland standards are prepared by mixing 1% barium chloride and 1% sulfuric acid, and the reaction between the barium chloride and the sulfuric acid produces a precipitate of barium sulfate, which is seen as the turbidity characteristics of the McFarland standard. So some common McFarland standards and the approximate bacterial concentration. So for the McFarland standard, we have 0.5, 1.0, 2.0, 3.0, and 4.0, and this is how they appear in terms of their turbidity. And this is the approximate bacterial concentration in CFU per mil. So for the 0.5 McFARLAND standard, the approximate bacterial concentration is 1.5 times 10 exponent 8. For the 1.0, the approximate bacterial concentration is 3.0 times 10 exponent 8. For the 2.0, we have 6.0 times 10 exponent 8. For the 3.0, we have 9.0 times 10 exponent 8. And for the 4.0 McFARLAND standard, the approximate bacterial concentration is 12.0 times 10 exponent 8 CFU per mil. And again, what you have to note is that in antibiotic susceptibility testing, we use the 0.5 McFARLAND standard with its approximate bacterial concentration to be 1.5 times 10 exponent 8 CFU per mil. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments or question below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.